This is B.Maheswaran, Assistant Professor, Department of Environmental Science, Virudhunagar, M.Sc. College, Virudhunagar. In this video tutorial, we are going to discuss about metabolism of xenobiotics phase 1 reactions. Before entering the topic, you must know that what is biotransformation. So biotransformation is a metabolic process that takes place mainly in the liver and helps to facilitate the excretion of both exogenous and as well as endogenous substances. A series of reactions may alter the chemical structure of these substances. The enzyme that catalyze these reactions can cause the substrate to become inactive, active, and even toxic material. The pathways of biotransformation which are divided into two, phase 1 and phase 2 reactions. These reactions may occur simultaneously or sequentially. Phase 1 which yield a polar water-soluble metabolite that is often still active. Many of the product in this phase also become substrate for phase 2 reactions. So hydroxylation by cytochrome P450, oxidative reactions, hydrolysis and reductions. So these are all the phase 1 reactions. Okay. Hydroxylation by cytochrome P450 is the chief reaction which is involved in phase 1. The responsible enzyme are so-called monooxygenases or cytochrome P450. It is estimated that there are some 57 cytochrome P450 genes present in humans. The reactions catalyzed by monooxygenases enzyme, so likely cytochrome P450. So here the reaction is written. So RH plus O2 plus NADB H plus H gives R bond OH plus H2O plus NADB. So the reaction one atom of oxygen enters R bond OH hydroxyl group and another one atom enters into water molecule. So H2, that is H2O. Okay. The set RH which represent a very wide variety of xenobiotics which include drugs, carcinogens, pesticides, petroleum products, pollutant, PCP like pollutant, endogenous compounds such as steroids, eicosanoids, fatty acids, retinoids are also a substrate for phase 2 reactions. These substrates are generally lipophilic in nature and are rendered more hydrophilic by hydroxylation reaction. Cytochrome P450 is considered the most versatile biocatalyst known to everyone actually. So in the above reaction that one atom enters into the oxygen and other one enters into the water molecule. So it also discussed. So here I cited one reaction. RH plus O2 gives R hyphen OH plus H2O. Here, reduced cytochrome P450 is converted to oxidized form of cytochrome P450. So the conversion occurs in this reaction. Okay. So in this reaction also toluene which is hydroxylated to pencil alcohol by mixed function of oxidase. So here toluene is converted to pencil alcohol. Here NADBH reduced oxygen is reduced here and forms NADB plus. Okay, cytochrome P450 is so named because the enzyme was discovered when it was noted that properties preparations of microsomes that had been chemically reduced and then expressed to carbon monoxide exhibited at a distinct peak at 415 nanometer. Microsomes contain fragments of the endoplasmic reticulum where much of the P450 content of cells is located. So among reasons are that these enzymes is the important one. Fact of approximately 50% of the common drugs using in human ingest or which are metabolized by isoforms of cytochrome P450. So these enzymes also act on various carcinogens and pollutant as well. The major cytochrome P450 is in drug metabolism or membranes of CYP1, CYP2 and CYP3 gene families. So here I listed some important points which concerning cytochrome P450. So it's like hemoglobin, they are hemoproteins, right? So they are widely distributed across among all the species. So they are present in highest number in living cells and erythrocytes, but are probably present in all tissues in human beings. So in liver and most of the other tissues, they are present mainly in the members of smooth endoplasmic reticulum, that is ACER, so which constitute part of the microsomal fraction when tissue is subjected to the subcellular fractionations. So in hepatic microsomes, cytochrome P450s can comprise as much of 20% of all the total protein present in the body. So P450s found in most of the tissues, though often at low amounts compared with liver. So where compared to adrenal, they are found in mitochondria as well as endoplasmic reticulum. There are various hydroxylase enzymes present in that organ which play an important role in cholesterol and steroid biosynthesis. So the mitochondrial cytochrome P450 system differs from the microsomal system in that of uses an NADPH linked flavoprotein and also adrenodoxin reductase and a non-heme iron sulfur protein adrenoxin. In addition to that, the specific P450 isoforms involved in steroid biosynthesis, which are generally much more restricted in their substrate specifically. So at least 6 isoforms of cytochrome P450 are present in the endoplasmic reticulum of human liver itself. So each with a wide and somewhat overlapping substrate specificities are also on both the xenobiotics and endogenous compounds. The genes for many isoforms P450 from both the humans as well as animal such as rat, likely rat, so have been isolated and studied in detail for prolonged years. The combination of there being a number of different numbers and each having a relatively wide specificity explains why the cytochrome P450 family, which can metabolize thousands of chemical compounds in daily life. Okay, so NADPH, not NADH. So here I mentioned NADPH. So it's the reaction of mechanism of cytochrome P450. The enzyme that uses NADPH cytochrome shown at the left hand side in the above reaction. So here the administration of phenylbarbital causes 3 to 4 fold increase in the amount of cytochrome P450. So lipids, also components of the cytochrome P450 system. The preferred lipid is phosphodicholine, so which is the major lipid found in membranes of endoplasmic reticulum. So oxidative reactions. So oxidative reaction, it may either aromatic or aliphatic hydroxylation. So the reaction may include sulfoxidation, epoxidation, N-oxidation. So phase 1, phase 2 reactions which are necessary to remove the chemical compounds from the environment. So for an example, benzene is converted to phenol through phase 1 reaction, oxidation reaction, and produced phenol is converted to phenyl sulfate at the end of conjugation, phase 2 conjugation process. So oxidation as well as detoxification of alcohol which is an important function of the liver. So the liver has play a main function of oxidation and detoxification of alcohol. Okay. So alcohol dehydrogenases and aldehyde dehydrogenases, these are two enzymes which is involved in oxidation and detoxification process. So here alcohol dehydrogenase, which oxidize alcohol to aldehyde group. So aldehyde dehydrogenase oxidize aldehyde to acid groups. So here the reaction may written as ethyl alcohol is converted to acetaldehyde by means of by the help of alcohol dehydrogenase enzyme. So and also acetaldehyde is converted to acetic acid and finally, so by the help of the enzyme called aldehyde dehydrogenase enzyme. So benzaldehyde also converted to benzoic acid by the help of aldehyde dehydrogenase. So oxidation process may produce a more toxic product. Here methanol at the end of forms formic acid, ethylene glycol, which is converted to oxalic acid at the end of the reaction, then a hydrolysis reaction. So certain therapeutic compounds which undergo hydrolysis process. So hydrolysis of the bond such as ester, glycoside and amide, which are important in the metabolism of xenobiotic compounds. So here H is incorporated into one fragment and a hydroxyl group is incorporated to another fragment. Okay, so aspirin, procaine, xylosine, acetylidine, so these are all the examples for hydrolysis reaction. So here aspirin which is hydrolyzed and finally produces salicylic acid plus acetic acid. So here H2O is mainly play a vital role to the conversion of salicylic acid plus acetic acid. So reduction reaction, so reduction is less common and less important than that of oxidation in every human being. So reductase is an enzyme which contains cytochrome P450 and flavoproteins. So reduction of nitro compounds which results in the toxification of their amines. So nitrobenzene is converted to aminobenzene. So here cytochrome P450 which play an vital role, and also the conversion of NADPH gives NADB. So here H is reduced to so H2O. So these are all reduction process. Thank you.